Welcome all to our talk in the History of Medicine series on RAS proteins in human disease. And this is by Frank McCormick. Frank McCormick is Diller Family Comprehensive Cancer Center, where he holds the David Wood Chair of Tumor Biology and Cancer Research at UCSF. I have known him really for scores of years now. Really, we worked on RAS oncogenes in the very early days, 1980s. So it's a singular honor to introduce you. So he also served as president of the American Association of Cancer Research, and before joining the UCSF faculty, he pursued cancer-related work with several Bay Area biotechnology firms and held positions with Cetus Corporation as director and Chiron Corporation, where he was vice president of research from 1991 to 1992. He really has moved between academia and the company and the fields with great ease. Now, in 1992, he founded Onyx Pharmaceuticals, a company that was dedicated to developing new cancer therapies, and served as its chief scientific officer until 1996. At Onyx Pharmaceuticals, he initiated and led the drug discovery efforts that led to the approval of sorafenib in 2005 for the treatment of renal cell cancer, and for liver cancer in 2007, and the approval of Onyx 015 in 2006 in China for treatment of nasopharyngeal cancers. So he discovered, among other things, he discovered the first RAF kinase and the CDK4 inhibitors entering the clinic. So you might have heard a lot about palbociclib. Indeed, that was discovered by Dr. McCormick. He also developed the first therapeutic agent designed to kill cells deficient in P53 or RB, and new approaches for targeting the RAS pathway, and to use systemic siRNAs and plasmid DNA as therapeutic platforms. So his lab continues to work on ways of treating breast cancer based on alterations in the RAS pathway. And more recently, he is now physically close to us since he has taken on leadership roles at Frederick National Labs for Cancer Research, overseeing an NCI-supported national effort to develop therapies against RAS-driven cancers. These cancers include, of course, most pancreatic tumors and colorectal and lung cancers, and are among the most difficult cancers to treat. So it's with great pleasure that I welcome you to talk to us about what else? RAS proteins in human disease.
Thank you very much. Thank you. It's great to be here. I think initially Sarah asked me to talk about the history of the discovery of oncogenes as a whole, but I thought that was a little too much to take on. So I'm going to give you a little bit of background of how RAS and other oncogenes were discovered back before most of you were born, and then give you my version of the history of discovery of RAS and its development as a drug target, and end up with some new data showing that actually RAS is a druggable protein, and there are actually drugs in the clinic now which target RAS directly, which is basically the first time I've been able to say that in a very long time. This is the protein that I'll be talking about, the RAS protein that we call the beating heart of signal transduction, a phrase I think coined by Hans Boss a while ago, when it was recognized that the RAS protein moves back and forth between two states, the off state and the on state, and this beating heart acts during signal transduction pretty much of all growth factors in all cells. So absolutely an essential part of signaling from growth factors into the inside parts of a cell, and the protein beautifully looks heart-shaped. So how did all this begin? Well, as I'm sure most of you who've read textbooks will remember that oncogenes and many tumor suppressors were identified through an analysis of viruses that cause cancer. And there were really two schools of research at that time, the DNA tumor virus group and the RNA tumor virus group. I won't say much about the DNA tumor virus people, although I was one of them, I must say, at the time. The DNA tumor viruses, very different lifestyle from the retroviruses. These viruses actually have to inactivate the RB protein to get into S phase to replicate viral DNA, and they trigger P53 in doing so they have to neutralize P53. So the DNA viruses encode proteins that neutralize RB and P53. And it was that understanding that led to a better understanding of those tumor suppressors. as part of the normal lifestyle of being a DNA virus, such as adenovirus or SV40 or papillomaviruses. So that led the field of DNA tumor viruses down towards the world of tumor suppressors. But the retroviral group really came up with all the genes that are activated in a dominant way as oncogenes. And again, this is textbook stuff, but you probably should all know at least that retroviruses, when they replicate in cells, they integrate, and when they move on, they often pick up pieces of cellular DNA which becomes integrated into the viral genome and can become mutated. And these pieces of cellular cDNAs essentially can then promote tumors. So these techniques were developed back in the 70s, again I'm sure before most of you were born, and led to the identification of pieces of cellular DNA which after mutation turn into oncogenes from proto-oncogenes. And these are a list of some of the genes that were identified by that technique. This essentially was a completely blind screen for genes that cause cancer, because none of the function of any of these proteins was understood at the time. This preceded any understanding at all of signal transduction, and it preceded the discovery of phosphotyrosine and tyrosine kinases, for example, by many years. So these people, most of whom are male, I have to point out, and Sarah's pointing that out, with one exception that I'll refer to, these dudes basically discovered viruses by taking retroviral stocks, injecting them into chickens or rats or other species, and then seeing that these retroviruses can promote tumors extremely aggressively and very efficiently. And they named each virus as it was purified and cloned after the disease itself, such as ERB, which is erythroblastosis virus type B, or the strain of the virus, the FIMS virus, which encodes the CSF1 receptor, stands for the feline sarcoidovirus of the McDonough strain. The KIT oncogene was named after the Sloan Kettering Institute for Research. The June oncogene, June is part of the Japanese word for 17, which is June Nana, because this was avian virus 17 and so on. So all of these names are completely sort of unrelated to any biology except for the diseases they cause, or the people who discovered them, or the institutes that they were discovered in. Now these are some of the people I just pulled off the web to give you a little test. I know most of you won't be familiar with any of these people because you're all too young, but you should know them because they're in the textbooks. Okay, here's the test. Anyone know who this guy is? Marina Barbasid. Chuck Schur, who discovered the CSF1 receptor and also discovered cyclin D1 and other cyclins. Hanafusa discovered. Sorry, yeah. Oh, I'm sorry. It's not? Oh, how do I point? Okay, I see. There we go. Sorry. Thanks for the... Okay, there we go. All right. That was an excuse because you don't recognize any of these people. Okay. So, Hanafusa, Sark, Peter Vogt, Baltimore, George van der Wood, and of course, Harold and Mike. Okay, and there are many others. Many of these people, by the way, were people who were trained to be medical students but went to the NCI to avoid the draft. And then they became pioneers in the discovery of these genes, either at NCI Bethesda or at Frederick, Maryland. There's one female amongst all these, and that is Jennifer Harvey, who discovered Harvey sarcoma virus, RAS, which is the foundation of the whole RAS field that I'll be talking about. But the interesting thing about this really is that this is, as I said, a completely blind screen for genes that cause cancer without any bias about which signaling pathways they should be looking at. But the pathways that they discovered, the pathway that they discovered, is really the same pathway, the growth factor, RAS, MAP kinase pathway. All of these genes, with a bit of a stretch, fit into this pathway. Now this is kind of remarkable because this screen could have found anything. It could have found genes in any signaling pathway, the hippo pathway or catenin or any pathway you like. But pretty much all the genes that are able to cause tumors acutely when injected directly into animals in an activated state are in the RTK signaling pathway. So this is the first clue, well it was the first clue that cancer can be initiated by activation of the RTK RAS pathway by mutant forms of these proteins that cannot be regulated properly. So for a long time, these names were just sort of referred to viruses but not proteins. But then the whole thing became clear when first Stu Aronson discovered that V-cis is related to PDGF growth factor, and that gave the idea that persistent growth factor signaling can cause cancer. That was the first clue. And then ERB was found to be related to the EGF receptor, which was sequenced by Julian Downward and Joshi Schlesinger. And then gradually things fell in place from there on out. Okay, so then the idea became that superactivation of growth factor signaling is sufficient to initiate cancer in these models. And by the way, the tumors that these viruses cause are typically leukemias and sarcomas, not carcinomas and other kinds of cancer. So these are tumor types which seem to be initiated by just one whack with a very strong oncogene. And by the way, drugs that target many of these oncoproteins have been the most successful drugs in cancer. So these are primary drivers of cancer, which really drive the bus in certain types of cancer. And they've been the subject of many therapeutic approaches, as you'll see from recognizing these genes. Okay, so phase one of this whole discovery process was often in vivo experiments where we see a single gene can promote tumors very quickly in certain strains of animals. And then things moved to the sort of the lab-based system for looking at oncogenes. And this made things much faster and easier and opened up the field of discovery of oncogenes in human tumor DNA. And this is the famous focus forming assay, which was, I think, first, the idea first, I think, came from Howard Temin and George Todaro and others. But the point of this assay is that you have a lawn of 3T3 cells here, which when they become confluent, they stop growing. But if one of these cells has an oncogene in it, it'll keep on growing and form foci which overgrow each other and form piles of disordered cells. That is the hallmark of an oncogene, okay? Not its ability to grow in low growth, without growth factors or anything like that. It's actually overgrowth and bad social behavior. That's the assay which all these oncogenes score in, and many more were discovered later on. So that's important to remember when you think about the mechanism of action of these genes, which I'll get back to later on. Now, the analysis of DNA from human tumors was facilitated by our ability to transfect DNA from one cell, from a tumor cell into cells in culture. And that transfection procedure was developed by Richard Axel and Mike Wigler, and really pioneered and exploited by many people, including Bob Weinberg and others, who showed that you could transfer drug resistance from a plasmid into cells and then select for them using DNA transfection, which is obviously now a trivial matter for all of you, but it was a big deal at the time because it enabled people to look for transforming genes by looking at their ability to form foci in cells after transfection of DNA extracted from tumors. Okay, and that opened up the whole field, and here you can see what a focus looks like. So that really changed everything. And using those kind of assays in 1992, 4 groups independently showed that DNA from a particular cell line, in this case the T24 EJ bladder carcinoma cell line, when dumped onto 3T3 cells, forms foci, and that when these foci are cloned out by some very clever tricks, the DNA that was capable of doing the transforming was identified as the T24 bladder cancer gene, which the driving gene is HRAS, the V12 mutation in HRAS. So these 4 groups simultaneously showed really the same thing, that this cell line contains a mutated form of the RAS gene identified by Jennifer Harvey many years earlier. So this assay discovered other oncogenes as well, including KRAS. This is from the work of Barbasid, who showed that if you look at do this transformation assay from a bunch of different cell lines, some of which you'll recognize. This is the HRAS in the T24, KRAS from this lung cancer cell line, and NRAS from this fibrosarcoma cell line, I think it is. And this assay also led to the discovery of of HER2/neu and other oncogenes that were derived from either tumors or from cell lines. So now we're looking at how these genes perform in a transformation assay, a quantitative transformation assay in cell culture. Now, the RAS protein was then sequenced from these genes and identified to be a single amino acid different from the wild type RAS protein. So this I remember very well when this happened, and it was discovered that a single base change in this DNA causes this transformation from a normal gene, which is involved in signaling in every cell, every normal cell, to an oncogene, which causes those gross foci in the focus-forming assay. So to me, that was a kind of epiphany, because I thought, well, if the mutant protein is only different by one amino acid, how hard will it be to fix it? So that was in 1984. We're still working on it. But really got my imagination, because it seemed like the whole of the sort of initiation of cancer process boiled down to just the difference in one amino acid between normal protein and a mutant protein, which I found really remarkable. When the protein was sequenced, and it looked unlike other oncogenes that had been identified. It did not appear to be related to other kinases which have been identified previously. So Ed Skolnick really led the charge here at the NIH that time, and showed that the RAS protein actually binds guanine nucleotides. It's not an ATP kinase as the other many oncogenes were, but actually bound to guanine nucleotides. And he found if you just label cell cultures expressing the viral HRAS gene with inorganic phosphate, as we used to do in the old days, a few millicures of P32 onto the cells, label them up nicely, and then immune precipitated the protein, he could see that the RAS protein is actually a phosphoprotein. In hindsight, this is kind of weird, because if you did this experiment today with a RAS gene from a human tumor, you would not see that band. Because it turns out, for really good luck for Ed Skolnick, the Harvey RAS oncogene has 2 amino acid changes compared with wild type, not just one. One is at codon 12, which activates it. The second one is at codon 59, which creates a threonine residue, which gets phosphorylated by the GTP that binds to the RAS protein. So it becomes a phosphoprotein by autophosphorylation. And now the RAS protein is a phosphoprotein which can be easily seen on a gel. That second hit on the threonine to create a phosphoprotein has never been seen in any human tumor DNA ever. So both Harvey sarcoma virus and Kirsten sarcoma virus, which was independently identified later, both of them have those 2 mutations. So we haven't yet figured out, or even tried to figure out, what the selective advantage is in having the second mutation when the first one is quite enough to turn the protein into an oncogene. Presumably the extra phosphorylation event gives it even further transforming power, but that hasn't really been validated. Anyway, it made it sort of relatively easy for Skolnick to realize that this is a GTP binding protein, and then that really changed the whole way we think about this class of oncogene. For one thing, it put the RAS P21 protein into a class of proteins which was quite well understood. It really looks like, very much like the bacterial elongation factors, which have the same G domain, the same GTP binding domain, and the signal transduction G proteins, such as G alpha, as shown here. So now we started to think about an oncogene as a G protein, not just as a kinase. Okay, now the GTP binding region of RAS is very similar to that of the G proteins and even bacterial elongation factors. In fact, this is the GTP molecule, and all the contacts are shown here around the protein that binds to the GTP molecule. And these are highly conserved between all members of the GTP binding family, from bacteria to humans and so on. So this is a very highly conserved motif. And so let's put the RAS into a zone which was already quite well understood. RAS is a GTP binding protein, it's a GTPase. It also opened up another whole box of really exciting genes, because once RAS was identified in 1982 or thereabouts, it was obvious it was part of a much bigger family. And this whole family was identified by looking at genes that were homologous to RAS, by using DNA low certainty hybridization and biochemistry and so on, to lead to the idea that this is part of a gigantic superfamily of small GTPases that regulate pretty much everything you can think of in cell biology. So I'm sure you're all familiar with the complexities of RAC and RO signaling in cell migration and actin cytoskeleton formation and ruffling and so on. The ARF proteins are involved in membrane trafficking and sorting in different compartments of the cell. The RAN proteins involved in nuclear export and import. RAG proteins regulate the Tor family of proteins, and so on. So this whole field basically exploded after RAS was discovered as an oncogene in 1982. We used to go to meetings of small GTPases where all of this group would get together and everybody would share their experiences from all kinds of different biological backgrounds. And also it was realized that these genes are highly conserved. So yeast has RAS genes and Rho and RAC and so on. So people working on model organisms were suddenly in the game because they could analyze the role of these proteins and the homologs and so on in all kinds of different biological systems, from yeast to flies to worms, and so the whole field kind of exploded at that point. So it was discovered again by Skolnick and co. that RAS proteins bind GTP. Then when these proteins were actually cloned as cDNAs and expressed in bacteria for the first time, we got a better idea of their biochemical properties. Now, this will seem trivial to you, but in those days, even cDNA cloning was really hard. And so it was a sort of tour de force to make a RAS protein in bacteria that was 1984 thereabouts. Remember, cloning really started in the 1980s, thereabouts. In 1980, 81, people started to be able to do cDNA cloning and express proteins in bacteria. So the whole field of cloning sort of exploded about the same time as the oncogene field. And that made it possible to do biochemistry on recombinant RAS proteins for the first time, instead of purified from cell extracts and whatnot. And when that was done, Jay Gibbs and Skolnick and friends showed that the wild-type HRAS protein had intrinsic GTPase activity, shown here, so it could hydrolyze GTP that bound to it, albeit very slowly. But the oncogenic version, the Val12 mutant, was impaired. This is about 4 to 5-fold reduction. So this, for a while, became the paradigm, that oncogenic RAS is active because it's weak at hydrolyzing GTP. It was known from the work on G proteins, from the signaling G proteins, the active form of these proteins is the GTP-bound form. The inactive form is GDP. So by hydrolyzing GTP to GDP, the protein turns itself off. And you can see from this graph that the mutant RAS protein turns itself off more slowly. And there we have what could be the sort of solution to why this point mutation makes this into an oncoprotein. It just slows down the GTPase rate. It's a sad side note on this. One of the intellectual drivers of the group at Merck at Skolnick, and actually the person who had the first idea to use retroviral therapy to treat the HIV epidemic at the time, was Irvin Siegel. And it gives me sort of goosebumps to think about it. It was so awful. He was killed on Pan Am Flight 103, coming back from a meeting in Europe, killed over Lockerbie. Sorry. Anyway, on we go. Sorry about that. So this is when I got into the game because I was working at a biotech company called Cetus, where we had figured out how to clone genes and express them in bacteria. And my friend, unfortunately also deceased, Alan Hall, was working in London, and he had cloned the NRAS gene. So NRAS wasn't identified as a retrovirus for some reason, but it was identified by transfection of DNA into 3T3 cells. But NRAS hadn't been characterized. So at a meeting in Spetses in Greece, actually, Alan asked if I would be able to help him clone and produce the recombinant NRAS protein to measure its biochemical properties. So I agreed to do that, and so we did just that. When we measured the GTPase rates of the mutant of the NRAS mutant, which happened to be the D12 mutant, the aspartate 12 mutant, we saw the GTPase activity was only 2 or 3-fold lower than wild type. Okay, so the Val12 is significantly lower, but AS12, which is a full-blown God-fearing oncogene, just as active in biological assays as the Val12 one, is barely different from wild type in its GTPase rate. So this, as we said in this paper, I think actually said the simple idea that mutant RAS proteins just have less GTPase was actually not right. We also knew that in biological assays developed by Carmen Birchmeyer when she was a postdoc in Mike Wigler's lab, where you inject RAS proteins into xenophus oocytes. You can measure their activity on a sort of biological unit per microgram of protein. The mutant RAS proteins are like 100-fold more active than wild type, not 2-fold or 3-fold. So we figured there was something wrong with the simple model that intrinsic GTPase activity was responsible for activation. So we decided to measure the GTPase rates in living cells. And we used Kalman's system of xenophus oocytes, where we loaded up RAS with screaming hot GTP, and then injected the RAS proteins into xenophus oocytes, and then measured the fate of the GTP over time in living cells. What we saw was that this is GTP and GDP. When we inject the wild-type protein into cells, within a matter of minutes, all the GTP is being converted to GDP. Okay? Whereas the mutant protein, the D12 or the V12, exactly the same, just stays with the same ratio of GTP to GDP pretty much forever. Obviously, the nucleotide comes off at some rate and gets diluted, but that's a very slow process as well. So the mutant protein is basically stuck in the GDP state. So we concluded there must be some cellular factor in xenophus oocytes which stimulates hydrolysis of GTP on wild-type RAS, but the mutant proteins are resistant to that factor. So we call the factor GAP because we found out it was a protein, and then we basically, at Frederick actually, we discussed the fact that we'd found a protein of unknown nature that stimulates hydrolysis of wild-type RAS GTP, but not the mutants. And this gave a much better quantitative reading of why the mutant proteins are so damn active compared with the wild-type. Then the race was on to clone the GAP cDNA by purification, old school purification, and then to get the cDNA clone. So we lost this race in my group to the group at Merck, who was much more organized. And they got a bit lucky. They chose the bovine brain as a source. We chose human placenta, which turned out to be complicated because there are two splice variants in human placenta, but they were just better than us, to be quite honest. So I sent my technician over to the maternity ward at Oakland Hospital with an ice bucket. So she waited alongside all the expectant fathers, for the birth of a baby, and then we got the placenta, and then we drove back to Emeryville and then purified the GAP activity from human placenta. But by what turns out to be luck also, both groups using bovine brain or human placenta came up with the same protein, which we call RAS-GAP, P120 RAS-GAP. So we defined the GAP domain, which is about 300 amino acids, and then these SH2, SH3 domains. So this was extremely interesting at that time. Again, this is another time when things were exploding in the field. Tony Pawson and his colleagues had identified SH2 domains and SH3 domains as signaling modules. So suddenly a whole range of proteins such as phospholipase T gamma and other GAPs and PI3 kinase and receptors were shown to have these modular signaling domains, SH2, SH3 domains, which can be plugged together like Lego to assemble signaling complexes. So RAS-GAP fell into this paradigm. So we and others show that RAS-GAP does indeed bind to growth factors through SH2 domains. So now it's starting to sort of all make sense a little bit. So this sort of scheme evolved in which RAS proteins cycled around from the inactive state through exchange factors, which I won't talk about today, to the GTP state. They turn on downstream pathways, and then GAPs come along, recruit to the receptor, and then turn RAS off again to go back to normal. And oncogenic mutants in RAS are resistant to the off switch from GAP and therefore stay in the GTP state. So this is sort of right, but not quite, as I'll show you. I have to mention also, this is not a comprehensive review of the whole history of RAS. That would take all day, which I'm happy to do, but you probably you may not be. In parallel to the work in mammalian cells and so on I've described, there are other parallel efforts to figure out this pathway using C. elegans and Drosophila, which have basically an identical RAS signaling pathway. And the other system that was used by Mike Wigler, who's probably one of the smartest people on the planet, he chose to use S. cerevisiae as a model system because yeast proteins have a RAS protein. Okay, yeast have a RAS protein. So he thought, why don't we do a whole genetics in the yeast, where it's much easier to do genetics in those days. And he developed some beautiful assays in which yeast would have superactive RAS by mutation, like Val12 RAS, causes heat shock sensitivity, because when you heat shock yeast, they stop growing and protect themselves, but mutant RAS just overrides that checkpoint and they just blast through and they die. Yeast with mutant RAS also don't bother to accumulate glycogen as a storage source. They just keep on growing. So even in the absence of a nutrient source, they just keep on growing. So you can stain yeast colonies with iodine, and you can see the mutants compared with the wild type, because they don't bother to store glycogen. So these are great assays for dissecting the pathway. And using these assays, Wigler identified exchange factors and gaps that regulate RAS in yeast. Human RAS will work just fine in yeast if you plug it in, even though it's never seen its downstream target. Because in yeast, it turns out by sheer bad luck for Mike, yeast activates adenyl cyclase RAS activates adenyl cyclase in yeast, okay, not RAF MAP kinase. So the same module of regulation is plugged into a completely different signaling pathway. So that was really bad luck. But everything else he discovered in yeast was very similar to the human situation, except the downstream pathway. And it turns out that this very same proteins, RAS, in different organisms performs different functions. So it activates cyclic AMP production in yeast, MAP kinase in POMB, but not through RAF, by a different MAP kinase. In dictyostelium, it activates PI3 kinase and mTOR, and then Drosophila and C. elegans is just as in mammalian cells, mostly RAF MAP kinase. So the whole machine of activating RAS and inactivating it by GEFs and GAPs has been extremely highly conserved, but the machine is plugged into different downstream modules in different organisms, which is kind of interesting. Again, we found that the gap we cloned binds to PDGF receptor when receptor is activated, and that led to the model in which the receptor activation involves, first of all, activation of RAS by recruitment of an exchange factor which kicks RAS into the active state, and then later on gap gets recruited and it turns RAS off again by stimulating conversion to the GTP, back to the GTP bound state. Okay? So that was the model. That basically, RAS is strictly involved in picking up signals from growth factors, and it's regulated by these exchange factors and gaps. However, I don't think it's quite that simple, because when you think back to the assay which led to the discovery of these RAS genes in the first place, the focus forming assay, that's not about growth factor independence or growth factor signaling per se. No amount of growth factors will make a normal cell form these foci and pile up on one another. They are very well behaved when they start to talk to each other. So what these oncogenes is doing is overcoming negative signals which normally prevent cells from actually proliferating when they're actually in contact with each other. And that phenomenon of overcoming contact inhibition was basically the sort of the foundation of the whole RAS, the whole oncogene field for many years. People have now moved on, and no one ever figured out what the basis of that actually is, even though it's the best predictor of an oncogene and oncogenic function. So as well as regulating growth factor signaling in response to upstream growth factors in this sort of pathway here, there are negative signals which get kicked in when cells actually form these tight junctions and form contact with each other, which also inhibit RAS. And oncogenic RAS overcomes these signals, as well as making growth factors less necessary. So these have not been well defined. So I just want to show one little vignette which suggests that there's some really interesting biology here. This is a review of cell-cell contacts and proteins that are involved in what this author calls is he in the audience? the secret handshake of cell-cell contact. And there are many different cell-cell contacts shown in this list here. But the one which I think is most relevant is the Efrain signaling pathway. So anyone know about Efrains? These are really cool, okay? Because the signaling comes not from a soluble ligand, but from another cell. So here's cell A, which has the Efrain ligand stuck to its plasma membrane, and the ligand is sticking out into the wind here. This is an adjacent cell, which has the tyrosine kinase signaling motif of the Efrain receptor inside the cell, and here's the extracellular receptor. So when these two cells come together, this ligand hooks up with this receptor, and you get a signal. But the signal is typically negative. Okay, so it's like a repulsive signal. So neurons use this kind of negative signal to guide the way around during development. It's like the cell finds itself in the wrong place, it gets like an electric shock and pulls back and stops. And that's used for guidance of cell positioning in many different aspects of cell biology. So the relevance to this discussion is work again came from Tony Pawson's group that showed that when you engage Efrains on this case neurites, you shut down MAP kinase signaling. And the reason for that, he believed then, and we've confirmed, is that Efrain receptors recruit P120 gap to the membrane. Okay, but in this case, the gap they recruit doesn't just turn off RAS, which Efrain's turned on itself, but it actually turns off RAS that's been activated by all other receptors in the cell as well. So when an Efrain receptor is engaged, it recruits gap, and it stays there long enough to turn off RAS that another receptor, such as VEGF or FGF, may have engaged on anywhere else in the cell. So it shuts down all RAS, not just locally, okay? So Efrains basically turn off MAP kinase signaling by recruiting gap to the membrane. Mutant RAS is resistant to gap, as I hope you all know. Cellular mutant RAS can't get negative signals from Efrains, okay? And as a result, you get uncontrolled proliferation. And you can see this in culture, but you can also see it in vivo. This is a disease called capillary malformation, arteriovenous malformation, in which arteries and veins sort of merge together and with terrible consequences. The genetics of this have been figured out, and typically this disease is caused by loss of function of P120 RAS gap, RAS-A1, or loss of function of Efrain B4. Either way, you get the same result. So this is what happens when cells of this particular lineage can't stop growing, and they keep on growing and form these muddled junctions. This is, I think, very nice genetic proof that this model is actually correct in this context. So then we modified the way we thought about gap a little bit and the role of RAS in cell-cell contact as well as proliferation. And then everything changed, for me anyway, when the NF1 gene was cloned in 1990. Okay, I'll admit it, I'd never heard of NF1 disease. I'm not a medical guy, but it's still stupid. But I did get a call from Ben Lewin, the editor of Cell, said, "Oh, Frank, the NF1 gene's been cloned and we want you to review the paper." So I acted like I knew what he was talking about and said, "Yeah, okay, I'm on it." He said, "No, you don't understand. The paper's already on the way to the printers. My only decision is to completely veto it. Otherwise, it'll get printed immediately." So I said, "Okay." And you can look at this snapshot here from Cell. This paper describing the cloning of the NF1 gene was received at Cell on July 24th, 1990, and appeared in the August issue. That's a fast turnaround. Yeah. This is a really big deal, though, because this disease has been known for many, many years. And as I'm sure I know many of you in the audience know, it has all kinds of complicated and awful phenotypes, including these benign dermal fibromas and more malignant tumors, including optical nerve tumors, Lisch nodules, bone formation problems, and learning problems. So half these cases are actually inherited from a parent who has a very mild form of the disease, but the child can be really severely affected because of unknown modifier genes which affect the penetrance. Very interesting stuff we don't understand yet. But the fact that this is the gene that is responsible for this disease is a RAS gap, as we showed. We followed up very quickly, I should say, with a paper we submitted in October the same year, and this came out in, I think, December the same year, showing that, in fact, the NF1 protein is a RAS gap that works on RAS itself. So this whole of this very complicated disease boils down to too much RAS signaling. Okay? And the reason that some of these lesions are focal is because this is a tumor suppressor. So affected individuals have one good allele and they lose the second one. Sorry, they lose the good allele and then they have both alleles are defective. Then they get focal growth, clonal growth, which forms these local tumors, benign tumors. So this complicated biology boils down to just too much active RAS. At least that's the model. But one way this made us sort of rethink the whole sort of RAS gap world was that the neurofibromin protein is actually very similar to the one that's expressed in yeast, the one that Mike Wigler had identified, all the way across the sequence of this protein. It's a huge protein. There's the gap domain. The rest of the protein is unknown territory. But the sort of stunning thing was that obviously yeast don't have tyrosine kinase signaling. So this protein must sense something else and regulate RAS in response to some other signal unrelated to standard RTK modular signaling. And we still haven't figured out what it is. And if anybody can figure out a way of doing it, please let me know. Come to my lab and you can enjoy a few years in California. Because the fact is, there are no other proteins that this NF1 binds to that really explain how it works. But when it's gone, then the cells have too much active RAS, and then the troubles begin. The protein itself is a if I can get that to work. It's supposed to be a movie. Oh, never mind. It is a gigantic homodimer, and we've solved structures of it in low resolution, and also bound to another protein called spread1, which loss of spread1 causes another disease called Legius syndrome. So a lot of good biochemistry on the protein, except we don't really understand how the whole thing functions in real life. Sorry?
It's moving now.
Is it? Oh, too late. I have no time for that kind of stuff. It moves, okay. Okay, so this field has been sort of stuck for quite a long time in trying to figure out how to deal with the NF1 disease from a therapeutic point of view, until very recently when another sort of goosebump-making discovery was made by Brigitte Wiedemann and colleagues at the NCI, and that is that children and adolescents who are treated with MEK inhibitors, such as sulimetinib, actually respond incredibly well to this kind of treatment. So this is an individual that's been treated, and you can see the effects of NF1 disease and the results of treatment with trametinib. So this, as just 2 days ago, got breakthrough therapy for treating these kids. The first time anything has worked in the neurofibroma condition. And this is obviously a subset of patients of a certain age group with a certain characteristics, but it's a huge step forward, and which is absolutely wonderful for the whole field. The NF1 loss or spread1 loss are part of a whole family of genes that we now call the RASopathies. These are typically germline mutations in activated parts of the RAS pathway. And these come in various types. So NF1 I mentioned, loss of NF1 or spread1 hyperactivates RAS, off we go. Costello syndrome is caused by germline mutation in RAS itself, HRAS, point mutations in HRAS, oncogenic HRAS mutations that cause all kinds of developmental problems, including learning disabilities and many other problems. Increased frequency of tumors, but that's not really the only problem that these kids have to deal with. CFC, also very similar, but it's caused by activating mutations in BRAF or in MEK, and gives a slightly different phenotype, but still an awful condition. Again, sporadic mutations, germline, but with devastating effects. And then Noonan syndrome, which is actually milder than the two previous ones, but is caused by one of many different genes. SHIP2 is the most common, and then SIBL, SOS, weak alleles of RAS, SHOC2, PP1C, RAF1, and several others. In fact, we have a paper in New Issue of Science showing 2 other genes involved in Noonan syndrome. So these are very weak alleles, and this disease can be inherited as well as sporadic. So we have a whole range of syndromes here caused by just too much RAS, just a little too much MAP kinase activity. And again, the hope is that using some of the better drugs that have been developed to treat cancer to shut down the MAP kinase pathway could be therapeutically useful in these conditions. One other one that came along more recently, is also a RASopathy, is the gene SYNGAP, which is identified as one of the genetic causes of autism. In fact, amongst the many genes associated with autism, this is one of the more frequent. So loss of function of SYNGAP causes autism. Now, in all the RASopathies I just mentioned, all these kids and adults have severe learning disabilities, which overlap with many of the features of autism. But really, the systemic effects of the RAS activation have been sort of more obvious. SYNGAP is only expressed in the brain. So loss of SYNGAP only affects the brain, obviously, and it seems like the major function is a phenotype related to autism. This is an extremely interesting area, suggesting that perhaps regulating MAP kinase signaling could actually have some benefit to people suffering from this type of autism. I'm quoting here a paper from your own Richard Huganier, who's a pioneer in SYNGAP activity. Okay, so very briefly, I just want to say a few words about targeting RAS cancers. First of all, the RAS genes are known to be frequently mutated in human cancer. The first big study that showed the frequency of RAS mutations was done here by Bert Vogelstein in collaboration with Hans Boss, and based on the discovery of Kari Mullis, who was actually in my group at CETUS, who discovered PCR. Whatever you read in the books about Kari Mullis, okay, a lot of it's true, but if he claims that he invented PCR because he thought of a better way of replicating DNA than God had, that's not true. What Kari Mullis was doing actually was trying to sequence single bases in the genome, focusing initially on the sickle cell allele and later on RAS mutations. So his idea was to lay down an oligo right by the codon of interest and extend it by one base and see which base gets incorporated to sequence that one base in the genome. And he tried that and got too much background, so he did it on both strands as a duplicate, and then had the brilliance to recognize the consequences of extending both strands to give you a template for exponential amplification. So that was discovered at CETUS in 1983, where I see he's working next to my lab. We had a huge machine called the Klenau Monster that did PCR reactions using Klenau DNA polymerase for every cycle before we came up with the thermostable version, and it was kind of clunky. But Hans Boss had the idea of streamlining this, so he developed a faster way of doing it and became known as the amplifying Dutchman. And he hooked up with Bert to show that using PCR you can actually detect RAS mutations relatively easily, and the frequencies now became much clearer. And now we know that RAS mutations occur in 95% plus of pancreas cancers and in each of these tumors shown here. So it seems in pancreas cancer and lung, most likely it's the major driver, and colorectal, not so sure because it's not the primary event, but obviously there's tremendous interest in targeting KRAS for therapy. Now here's a paper which I thought was a classic, came from Sarah. I often quote this and make my students read this paper. This is actually, I pulled out the wrong one, sorry here, but it's one of a series of papers by Sarah in which she had done a really great experiment of injecting carcinogens into pregnant buffalo rats. And then these rats give birth to pups, and the pups eventually get tumors, in most cases mammary tumors. And the brilliant part of it, like this is, that the mutations that activate the RAS, HRAS in these mammary glands are actually the same, completely consistent with the mode of action of the carcinogen in the parental rat. Okay? So that says a lot about the mutations being most likely the primary initiating event. Also the fact that the pups grow up quite a long time before they get tumors means that the mutations must have been in the stem cells. Also, they don't get tumors until they go through puberty, and then a second sort of environmental hit kicks them into gear and they become tumors. So if she castrated them, they don't get mammary tumors, they get other tumors later in life. So this tells a lot about the initiating role of RAS in tumors and how it's the codependence on other signals and the fact that it most likely occurs in stem cells. Moving forward now, we can now look at the frequency of RAS mutations in human tumors, and we can actually ask on a web-based system, the DepMap, which I use a lot, what is the dependency of RAS-containing cell lines for continued expression of RAS? So if you haven't used DepMap, I should look it up. You should look it up and have a look. This top line here, which you probably can't read from there, this line, these are all the cell lines in this analysis that have mutations in pancreas cancer. The brown dots are pancreatic cancer. So anything to the left of this line means the tumor cell depends on that gene, in this case KRAS. So you knock it down by CRISPR or shRNA, you see a shift to the left. So all of these cells are dependent on continued expression of KRAS. But you can see there's a big variation in expression across all of these different cell lines. But for the most part, they stay dependent on KRAS for proliferation in cell culture. So that means that if it means anything at all, probably targeting RAS is a really good idea, even as a late-stage event in tumors. But the idea of targeting RAS has obviously been around for a long while, and the first attempts to target RAS, which some of you will remember maybe with pain, was the attempt to block RAS using farnesyltransferase inhibitors. So it was discovered by Chris Marshall and John Hancock and Doug Lowy and others that RAS proteins are farnesylated, and they have to be farnesylated to be active. And this farnesylation reaction happens at the C-terminus of the protein and plugs it into the plasma membrane. The enzyme that transfers the farnesyl group to RAS is called farnesyltransferase, so the whole biopharma industry saw this as an opportunity to inhibit RAS. This is back in the 80s when we didn't even know the extent of RAS involvement in cancer. So they all jumped in and they all developed very good farnesyltransferase inhibitors. Unfortunately, they failed. And that's because, unknown to the field at the time, when you inhibit farnesylation of RAS proteins, the two forms of RAS which play the biggest role in human cancer can undergo a backup system of general transferase. And that rescues them and allows them to keep growing. We didn't know that when these drugs went into the clinic, I don't think. I think it was discovered as part of this process of understanding why they hadn't worked as well as we'd hoped. Unfortunately, a lot of the preclinical work on RAS as targets was done on HRAS because that was the first RAS gene identified, and mouse models were made with HRAS, and everybody cloned and used HRAS proteins. So the biochemistry and biology of RAS was largely based on HRAS. But of course, we now know KRAS is the major player. So we think these drugs failed because of this backup system which basically allows KRAS and NRAS to keep going when HRAS is inhibited. However, now we can target tumors that have HRAS specifically. Couldn't do that in the 80s. Now we can pre-select patients that have mutations in HRAS. In collaboration with a group now called Cure Oncology, Kay van Shokat and I persuaded Janssen to license tipifarnib, one of the farnesyltransferase inhibitors which is still in play, to test tipifarnib in tumors pre-selected for having HRAS mutations. And that is 5% of bladder cancer, about 5% of thyroid cancer, and about the same of squamous cell carcinoma of the head and neck. So this clinical trial is now in a phase 3 pivotal study based on very good response rates in phase 2 as a single agent. So patients with HRAS-driven tumors do appear to respond to farnesyltransferase inhibitors. Okay? Now it's not proven yet, but it looks good so far, and we'll know fairly soon. Obviously great news for the patients who have HRAS-driven tumors, also great news for the field as a whole, because it means that in some cases just targeting RAS with a single agent drug might be sufficient to have an effect without having to do combinations, although that will happen later, I'm sure. Anyway, so that says to me anyway that HRAS is a good target, and by extrapolation most likely N and K will be as well. Unfortunately, the surface of the RAS protein shown here in its inactive and active states doesn't have many deep pockets where a small molecule can bind. So most drug companies looked at this and said, we're not going to touch this. There's no way we can get a small molecule to bind to this surface. And they gave up on RAS pretty much after the farnesyltransferase disappointment in the 80s. And then moved on to downstream. So this is what the pathway looked like in 1992. That happens to be when I started Onyx. So we said, okay, we'll give up on RAS, but we'll just target RAF. It's a kinase, it's easy tractable target. What could possibly go wrong with that? Well, it turned out pretty much everything as far as RAS cancers go, because they did not work. We did develop sorafenib, as Sarah said, which is actually pretty potent against RAF, but it didn't have any benefit in RAS-driven tumors. In the phase 1, we saw some benefit in liver cancer and in kidney cancer, but those are not tumors which have mutations in RAS. So all the RAS-driven tumors are basically struck out completely, really. Now, we knew something was weird going on here, because in patients treated with sorafenib, you start to see these little tumors appearing on the skin. These are keratocanthomas, or squamous cell carcinomas of the skin, caused by treatment with the RAF inhibitor. So we thought initially this is some weird effect of sorafenib, but it turned out that vemurafenib and all other RAF inhibitors that went to the clinic have the same thing. RAF inhibitors activate RAF, not what we had in mind. This summarizes this a ton of work in this nice study from Neil Rosen. This is a cell line that has very little phospho-MEC, which is the substrate, until you add increasing concentrations of different RAF inhibitors, and they increase the level of phospho-MEC. Therefore, they've activated RAF. And you get a kind of bell-shaped curve. Now, this led to a whole flurry of Cell papers and Nature papers explaining this weird phenomenon. And bottom line is, RAF is a dimer. If you inhibit one half of a dimer, it hyperactivates the other half. So that makes things worse until you shut them both down. And by that time, the drug has to be used at too high a concentration. So that didn't work for reasons which nobody expected. So then the next thing was, well, let's go down to MEK and see if that works. Now we get into the sort of mess of the signaling pathway as we now see it, not just a linear pathway down the MAP kinase pathway, but RAS activates all kinds of other pathways with all these feedback loops. And essentially, any attempt to poke at this pathway in a RAS transformed cell has failed because of feedback or compensation or redundancy and so on. So shutting down the network has not worked, down in RAS cancers anyway. Let's skip that. So until about 5 years ago, we're stuck with the idea that we can't treat RAS directly. RAS tumors don't respond to any therapies. In fact, as shown for this survival study for alotinib plus chemotherapy in non-small cell lung cancer, patients with mutant RAS do worse, particularly when treated with the EGF receptor inhibitor. So the major contribution to RAS in the clinical world at this point was excluding RAS mutant patients from treatment, from lung cancer and colorectal cancer, and also in melanoma later. So that's obviously not a very satisfactory outcome. But then things changed for the better when Keyvan Shokat at UCSF had a brilliant idea. Keyvan recognized that in non-small cell lung cancer, a very predominant mutation is the G12C mutation. And that's because the conversion of the codon for glycine to cysteine is the hallmark of exposure to tobacco smoke. Okay, so it's very frequent in smokers in non-small cell lung cancer, less in the other tumors. But G12C provides a chemical opportunity because the cysteine group is a reactive side group that can be attacked with electrophilic chemicals, unlike glycine itself, obviously, and other mutations. So Keyvan had the idea that a chemical could be found that binds to the cysteine residue and would then block RAS function. And it would be totally specific for the mutant, obviously, because there's no cysteine at that site in the wild-type protein, and would shut down the mutant protein. And indeed, that's what's happened. So he published this paper, and then there was like a stampede from the biopharmaceutical world, and actually Amgen won that race. They were in the clinic first with the first G12C inhibitor at the end of last year, and the biotech company Mirati was second a few months later. And we hear that Sanofi and Wellspring and many other companies have different versions of G12C. Because once you get the idea, it's kind of easy. You know? It's a very small pocket, and you're attacking a cysteine, there's not much room to maneuver. I would guess that one of these compounds, or more of them hopefully, will be active against the G12C-driven tumors, and that'll be another point on the board against RAS cancers, along with the HRAS I just described. Okay, so that's good for G12C, but we're running out of time, so I'll skip ahead. But not for G12D. The aspartate group is not a reactive side group like cysteine, and valine obviously not either. So now everybody's scratching their heads trying to find ways of attacking the other mutants selectively. And there are lots of possibilities, which I'll just summarize a couple here to skip on ahead of time. One is to prevent processing of KRAS. This is one of the things we're doing at the Frederick National Lab, to find a farnesyltransferase equivalent for KRAS by finding a chemical that reacts with the cysteine where KRAS is normally processed. We've found compounds that do that, and they wipe out KRAS in cells and have no effect on cells in which RAS is mutated at that site and rescued by myrcylation at the immunoterminus. So we have very nice sort of early stage compounds that hit KRAS at the C-terminus and affect processing. And it turns out, here's the cysteine at the C-terminus. It turns out if the movie works, which it probably won't unless I get rid of this. Nope. Sorry. How do I stop this? I don't know. Oh, okay. Yeah. So we found out that the tail actually folds back on the G domain, creates a pocket, and we can now covalently hit the cysteine and then block KRAS function. So that's one approach which we are developing kind of aggressively. Another approach, the last thing I'll mention, is based on the observation that in the G domain of KRAS, there's a histidine residue which is unique to KRAS, only found in KRAS and not HRAS or NRAS. So a drug that targets this histidine here could have effects on selectively blocking KRAS function without affecting HRAS and NRAS. And that's another whole strategy, because ideally targeting the mutant allele is great, but if you can't do that for the valine or the aspartate mutants, targeting KRAS period is a pretty viable strategy, because we know from Mariana Barbas's work again that if you wipe out KRAS in an adult mouse, they are fine. They live for about at least a year or so without any obvious side effects, because HRAS and NRAS can back them up in normal tissue. So obliterating KRAS is going to be effective in a KRAS tumors as long as you don't hit H and NRAS as well. So this approach here would be one way to get at that. And again, there's a whole series of different approaches which I don't have time to talk about. But the sort of bottom line is that, that if we turn the clock back to the 70s when the RAS genes were identified, we could recognize immediately the power of the oncogenes to cause tumors in cells, in vivo and in cells. We figured out a lot about the regulation and the pathways that RAS proteins regulate. Still don't have a very clear idea exactly how it causes cancer and how it works at different stages, but we are now at a point where we can see therapeutic intervention is possible. Just actually the last RAS meeting we held in San Diego last December, there was a whole session on RAS therapies, and that's the first time that's ever happened. So now we've got MEK inhibitors for NF1, farnesyltransferases for HRAS, and a whole range of small molecules targeting KRAS in different ways. So in the period of that time, I think we've come to the point where we can start to see that the RAS oncogenes may have met their end. And with that, I'll stop and take questions. Thank you very much. Thank you. Do we have time for questions? I'll be happy to take questions. No? No questions? Okay.
But may I take a moment to just give you a token of our appreciation? Yes.
Oh.
Please. If you would come on over here.
Okay.
This is for you.
Thank you. Okay, thank you. Wow, that's nice. Okay.
Hi, that was fantastic. Frank, really nice job.
Hi, great to see you. Yeah.
Yeah, nice to see you.
How's
All right.
life?
Very good, thank you.